12. Mineral nutrition. The basic needs of all living organisms are essentially the same. They require macromolecules such as carbohydrates, proteins, and fats, and water and minerals for their growth and development. This chapter focuses mainly on inorganic plant nutrition, wherein you will be studying the methods to identify elements essential to growth and development of plants and the criteria for establishing the essentially. You will also study the role of essential elements, the major deficiency symptoms, and the mechanism of absorption of these essential elements. The chapter also introduces you briefly to the significance and mechanism of biological nitrogen fixation. 12.1 Methods to study the mineral requirements of plants. In 1860, Julius von Sachs, a prominent German botanist, demonstrated for the first time that the plants could be grown to the maturity in a defined nutrient solution in complete absence of soil. This technique of growing plants in a nutrient solution is known as hydroponics. Since then, a number of improvised methods have been employed to try and determine the mineral nutrition essential for plants. The essence of all these methods involved the culture of plants in a soil free, defined mineral nutrition. These methods require purified water and mineral nutrient salts. Can you explain why is this so essential? After a series of experiments in which the roots of the plants were immersed in a nutrient solution and wherein an element was added, substituted, removed, or given in a varied concentration, a mineral solution suitable for the plant growth was obtained. By this method, essential elements were identified and their deficiency symptoms discovered. Hydroponics have been successfully employed as a technique for the commercial production of vegetables such as tomato, seedless cucumber, and lettuce. It must be emphasized that the nutrient solution must be adequately aerated to obtain the optimum growth. What would happen if solutions were poorly aerated? Diagrammatic views of the hydroponic technique is the given in figure 12.1 and 12.2. 12.2 Essential mineral elements. Most of the mineral present in soil can enter plants through roots. In fact, more than the 60 elements of the 105 discovered so far are found in different plants. Some plant species accumulate selenium, some other gold, while some plants growing near nuclear test sites take up radioactive strontium. These are techniques that are able to detect the mineral even at a low concentration. The question is whether all the diverse mineral elements present in a plant, for example, gold and selenium, as mentioned above, are really necessary for plants? How do we decide what is essential for plants and what is not? 12.2.2 Criteria for essentiality. The criteria for essentiality of an element are given below. A. The element must be absolutely necessary for supporting normal growth and reproduction. In the absence of the element, the plants do not complete with their life cycle or set the seeds. B. The requirement of the element must be specific and not replaceable by another element. In other words, deficiency of any one element cannot be met by supplying some other element. C. The element must be directly involved in the metabolism of the plant. Based upon the above criteria, only a few elements have been found to be absolutely essential for plant growth and metabolism. These elements are further divided into two broad categories based on the quantitative requirements. The very first is macronutrients and the second is micronutrients. Macronutrients are generally present in plant tissues in large amounts, in excess of 10 millimole per kg of dry matter. The macronutrients include carbon, hydrogen, oxygen, phosphorus, sulfur, potassium, calcium, and magnesium. Of these, carbon, hydrogen, and oxygen are mainly obtained from carbon dioxide and water, while the others are absorbed from the soil as mineral nutrition. Micronutrients or trace elements are needed in very small amounts. These include iron, manganese, copper, molybdenum, zinc, boron, chlorine, and nickel. In addition to the 17 essential elements named above, there are some beneficial elements such as sodium, silicon, cobalt, and selenium. They are required by higher plants. Essential elements can also be grouped into four broad categories on the basis of these diverse functions. These categories are the very first, essential elements are components of biomolecules and hence structural elements of cell, example carbon, hydrogen, oxygen and nitrogen. Second, essential elements that are components of energy related chemical compounds in plants, example magnesium in chlorophyll and phosphorus in ATP. Third, essential elements that activate or inhibit enzymes, for example magnesium is an activator for both ribulose bisphosphate carboxylase oxygenase and phosphoenol pyruvate carboxylase, both of which are critical enzymes in photosynthetic carbon fixation. Zinc is an activator of alcohol dehydrogenase and molybdenum of nitrogenase during nitrogen metabolism. Can you name a few more elements that fall in this category? For this, you will need to recollect some of the biochemical pathways you have studied earlier. Fourth, some essential elements can alter the osmotic potential of a cell. Potassium plays an important role in the opening and the closing of stomata. You may recall the role of minerals as solutes in determining the water potential of a cell. 12.2.2 Role of macro and micronutrients. Essential elements perform several functions. They participate in various metabolic processes in the plant cell, such as permeability of a cell membrane, maintenance of osmotic concentration of cell sap, electron transport system, buffering action, enzymatic activity, and act as a major constituents of macromolecules and coenzymes. Various forms and functions of essential nutrient elements are given below. Nitrogen. This is the essential nutrient element required by plants in greatest amount. It is absorbed mainly as NO3 minus, though some are also taken up as NO2 minus or NH4 plus. Nitrogen is required by all parts of plant, particularly the meristematic tissues and the metabolically active cells. Nitrogen is one of the major constituents of proteins, nucleic acids, vitamins, and hormones. Phosphorus. Phosphorus is absorbed by the plants from the soil in the form of phosphate ions. Phosphorus is a constituent of cell membranes, certain proteins, all nucleic acids and nucleotides, and is required for the phosphorylation reaction. Potassium. It is absorbed as potassium ion. In plants, this is required in more abundant quantities in the meristematic tissues, buds, leaves, and root tips. Potassium helps to maintain an anion cation balance in the cells and is involved in protein synthesis, opening and closing of stomata, activation of enzymes, and in the maintenance of turgidity of cells. Calcium plays important role. Alright, wait. Calcium. Plants absorb calcium from the soil in the form of calcium ions. Calcium is required by the meristematic and differentiating tissues. During cell division, it is used in the synthesis of cell wall, particularly as calcium pectate in the middle lamella. It is also needed during the formation of mitotic spindle. It accumulates in older leaves. It is involved in the normal functioning of the cell membranes. It activates certain enzymes and plays an important role in regulating metabolic activities. Magnesium. It is absorbed by plants in the form of divalent magnesium. It activates the enzymes of respiration, photosynthesis, and are involved in the synthesis of DNA and RNA. Magnesium is a constituent of the ring structure of chlorophyll and helps to maintain the ribosome structure. Sulfur. Plants obtain sulfur in the form of sulfate. Sulfur is present in two amino acids, cysteine and methionine, and is the main constituent of several coenzymes, vitamins like thiamine, biotin, coenzyme A, and ferredoxin. Iron. Plants obtain iron in the form of ferric ions. It is required in larger amounts in comparison to other micronutrients. It is an important constituent of proteins involved in the transfer of electrons like ferredoxin and cytochromes. It is reversibly oxidized from Fe2 positive to Fe3 positive during electron transfer. It activates catalyze enzyme and is essential for the formation of chlorophyll. Manganese. It is absorbed in the form of manganese ions. It activates many enzymes involved in photosynthesis, respiration, and nitrogen metabolism. The best defined function of the manganese is in the splitting of water to liberate oxygen during photosynthesis. Zinc. Plants obtain zinc as zinc ions. It activates various enzymes, especially carboxylases. It is also needed in the synthesis of oxygen, copper. It is observed like cupric ions. It is essential for the overall metabolism in plants. Like iron, it is associated with certain enzymes involved in redox reaction and is reversibly oxidized from Cu plus to Cu2 plus. Boron. It is absorbed as BO33 minus or B4072 minus. Boron is required for uptake and utilization of calcium, membrane functioning, pollen germination, cell elongation, cell differentiation, and carbohydrate translocation. Molybdenum. Plants obtain it in the form of molybdenate ions. It is a component of several enzymes, including nitrogenase and nitrate reductase, both of which participate in the nitrogen metabolism. Chlorine. It is observed in the form of chloride ion. Along with the sodium and potassium, it helps in determining the solute concentration and the anion cation balance in cells. It is essential for the water splitting reaction in photosynthesis, a reaction that leads to oxygen evolution. 12.2.3 Deficiency symptoms of essential elements. Whenever the supply of an essential elements become limited, plant growth is retarded. The concentration of the essential element below which plant growth is retarded is termed as critical concentration. The element is said to be deficient when present below the critical concentration. Since each element has one or more specific structural or functional role in plants, in the absence of any particular element, plants show certain morphological changes. These morphological changes are indicative of certain elements' deficiencies and are called deficiency symptoms. The deficiency symptoms vary from element to element and they disappear when the deficient mineral nutrient is provided to the plant. However, if deprivation continues, it may eventually lead to death of the plant. The parts of the plant that show the deficiency symptoms also depend on the mobility of the element in the plant. For elements that are actively mobilized within the plants and exported to the young developing tissues, the deficiency symptoms tend to appear first in the older tissues. For example, the deficiency symptoms of nitrogen, potassium, and magnesium are visible first in the senescent leaves. In the older leaves, the biomolecules containing these elements are broken down, making these elements available for mobilizing to younger leaves. The deficiency symptoms tend to appear first in the young tissues whenever the elements are relatively immobile and are not transported out of the mature organs. For example, elements like sulfur and calcium are a part of structural component of the cell and hence are not easily released. This aspect of mineral nutrition of plant is of great significance and importance of agriculture and horticulture. The kind of deficiency symptoms shown in the plant include chlorosis, necrosis, stunted plant growth, premature fall of leaves and buds, and inhibition of cell division. Chlorosis is the loss of chlorophyll leading to yellowing in leaves. The symptom is caused by the deficiency of elements like nitrogen, potassium, magnesium, sulfur, iron, magnesium, zinc, and molybdenum. Likewise, necrosis or death of tissue, particularly leaf tissue, is due to deficiency of calcium, magnesium, copper, potassium, lack or low level of nitrogen, potassium, sulfur, molybdenum causes an inhibition of cell division. Some elements like nitrogen, sulfur, molybdenum delay flowering if there is a concentration in the plant is low. You can see from the above that the deficiency of any elements can cause multiple symptoms and that the same symptoms can be caused by the deficiency of one of the several different elements. Hence, to identify the deficient element, one has to study all the symptoms developed in the various parts of the plant and compare them with the available standard tables. We must also be aware that the different plants also respond differently to deficiency of the same element. 12.2.4 Toxicity of micronutrients. The requirement of micronutrients is always in low amounts, while their moderate decrease causes the deficiency symptoms and a moderate increase causes toxicity. In other words, there is a narrow range of concentration at which the elements are optimum. Any mineral ion concentration in tissues that reduces the dry weight of the tissues by about 10% is considered toxic. Okay. Such critical concentration vary widely among different micronutrients. The toxicity symptoms and difficult to identify. Toxicity levels for any elements also vary for different plants. Many a times excess of an element may inhibit the uptake of other element. For example, the prominent symptom of manganese toxicity is the appearance of brown spots surrounded by chlorotic veins. It is important to know that the manganese competes with the iron and magnesium for uptake and with magnesium for binding with enzymes. Manganese also inhibit calcium translocation in shoot apex. Therefore, excess of manganese may in fact include deficiency of iron, magnesium, and calcium. Thus, what appears as symptoms of manganese toxicity may actually be the deficiency symptoms of iron, magnesium, and calcium. Can this knowledge be of some importance to be a farmer, a gardener, or even for you in a kitchen garden? 12.3 Mechanism or absorption of elements. Much of the studies on mechanism of absorption of elements by plants has carried out in isolated cells, tissues, or organ. They reveal that the plant process of absorption can be demarcated into two main phases. In the first phase, an initial rapid uptake of ions into the free space or outer space of cells, the apoplast is passive. In the second phase of the uptake, the ions are taken into slowly into the inner space, the symplast of the cells. The passive movement of the ions into apoplast usually occurs through the ion channels, and the transmembrane proteins the function as the selective ports. On the other hand, the entry or the exit of ions to and from the symplast requires the expenditure of metabolic energy, which is an active process. The movement of ions is usually called flux. The inward movement into the cells is influx, and the outward movement is called efflux. You have read the aspects of mineral nutrient uptake and translocation plants in chapter number 11. 12.4 Translocation of solutes. Mineral salts are translocated through xylem along with the ascending stream of water, which is pulled up through the plant by the transpirational pull. Analysis of xylem sap show that the presence of mineral salts in it. Use of radioisotopes of mineral elements also substantiate the view that they are transported through the xylem. You have already discussed the movement of water in xylem in chapter number 11. 12.5 Soil as reservoir of essential elements. Majority of nutrients that are essential for the growth and development of plants become available to the roots due to weathering and breakdown of rocks. These processes enrich the soil, which dissolve ions and inorganic salts. Since they are derived from the rock minerals, their role is in plant nutrition is referred to as mineral nutrition. Soil consists of a wide variety of substances. Soil not only supplies minerals but also harbors nitrogen fixing bacteria. Other microbes hold water, supplies air to the roots, and act as a matrix that stabilizes the plant. Since deficiency of essential mineral affect the crop yield, there is often a need for supplying them through fertilizers. Both the macro like nitrogen, phosphorus, potassium, sulfur, etc., and micronutrients that is copper, zinc, iron, manganese, etc., form components of fertilizers and are applied as per need. 12.6 Metabolism of nitrogen. 12.6.1 Nitrogen cycle. Apart from carbon, hydrogen, and oxygen, nitrogen is the most prevalent element in living organisms. Nitrogen is a constituent of amino acids, proteins, hormones, chlorophylls, and many of the vitamins. Plants compete with the microbe for the limited nitrogen that is available in soil. Thus, nitrogen is a limiting nutrient for both natural and agriculture ecosystems. Nitrogen existing as two nitrogen atoms joined by a very strong triple covalent bond. The process of conversion of nitrogen to ammonia is termed as nitrogen fixation. In nature, lightning and ultraviolet radiation provide enough energy to convert nitrogen to nitrogen oxides. Industrial combustions, forest fire, automobile exhaust, and power generating stations are also sources of atmospheric nitrogen oxides. Decomposition of organic nitrogen of dead plants and animals into ammonia is called ammonification. Some of this ammonia volatilizes and re-enters the atmosphere, but most of it is converted into nitrate by soil bacteria in the following steps. Ammonia is first oxidized to nitrite by the bacteria Nitrosomonas and Nitrosococcus. The nitrate is further oxidized to nitrate with the help of bacterium Nitrobacter. These steps are called nitrification. These nitrifying bacteria are chemoautotrophs. The nitrate thus formed is absorbed by the plants and is transported to the leaves. In leaves, it is reduced to form ammonia that finally forms the amine group of amino acids. Nitrate present in the soil is also reduced to nitrogen by the process of denitrification. Denitrification is carried by bacteria Pseudomonas and Theobacillus. 12.6.2 Biological nitrogen fixation. Very few living organisms can utilize the nitrogen in the form of nitrogen N2 that is available abundantly in the air. Only certain prokaryotic species are capable of fixing nitrogen. Reduction of nitrogen to ammonia by living organisms is called biological nitrogen fixation. The enzyme nitrogenase, which is capable of nitrogen reduction, is present exclusively in prokaryotes. Such microbes are called nitrogen fixers. The nitrogen fixing microbes could be free living or symbiotic. Examples of free living by nitrogen fixing aerobic microbes are Azotobacter and Bigenicia, while Rhodospirillum is anaerobic and free living. In addition, a number of cyanobacteria such as Anabaena and Nostoc are also free living nitrogen fixers. Symbiotic biological nitrogen fixation. Several types of symbiotic biological nitrogen fixing associations are known. The most prominent among them is legume bacteria relationship. Species of root-shaped rhizobium has such relationship with the roots of several legumes such as alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean, clover bean, etc. The most common association on the roots is as nodules. These nodules are small outgrowths on the roots. The microfrankia also produce nitrogen fixing nodules on the roots of the non-leguminous plants, example Ellis. Both rhizobium and frankia are free living in the soil, but our symbionts can fix atmospheric nitrogen. Uproot any of the plant common like a common pulse just before flowering. You will see near spherical outgrowths on the roots. These are called nodules. If you cut through them, you will notice that the central portion is red or pink. What makes the nodules pink? This is due to the presence of leguminous hemoglobin or leg hemoglobin. Nodule formation. Nodule formation involves a sequence of multiple interaction between the rhizobium and roots of the host plant. Principal stages in the nodule formation are summarized as follows. Rhizobia multiply and colonize the surroundings of roots and get attached to the epidermal and root hair cells. The root hairs curl and the bacteria invade the root hairs. An infection thread is produced carrying the bacteria into the cortex of the root, where they initiate the nodule formation in the cortex of the root. Then the bacteria are released from the thread into the cell, which leads to the differentiation of the specialized nitrogen fixing cells. The nodule thus formed establishes a direct vascular connection with the host for exchange of nutrients. These events are depicted in figure 12.4. The nodule contains all the necessary biochemical components such as enzyme nitrogenase and leg hemoglobin. The enzyme nitrogenase is like molybdenum and iron protein and catalyzes the conversion of atmospheric nitrogen to ammonia. The first stable product of nitrogen fixation. The reaction is as follows. The enzyme nitrogenase is highly selective to the molecular oxygen. It requires anaerobic condition. The nodules have adaptation that ensures the enzyme is protected from oxygen. To protect these enzymes, the nodule contain an oxygen scavenger called hemoglobin. It is interesting to know that these microbes live as aerobes under free living condition. But during the nitrogen fixing events, they become anaerobic. You must have noticed in the above reaction that the ammonia synthesizes by nitrogenase requires a very high input of energy, that is 8 ATP for each NH3 produced. The energy required thus is obtained from the respiration of the host cell. Fate of ammonia. At physiological pH, the ammonia is protonated to form NH4 positive, that is ammonium ion, while most of the plants can assimilate nitrate as well as ammonium ions. The latter is quite toxic to the plants and hence cannot accumulate in them. Let us now see how the ammonium is used to synthesize amino acids in plant. These are the two main ways in which this can take place. First, reductive amination. In these processes, ammonia reacts with alpha-ketoglutarate acid and forms glutamic acid as indicated in the equation given below. Second is transamination. It involves the transfer of amino group from one amino acid to the keto group of a keto acid. Glutamic acid is the main amino acid from which the transfer of NH2, the amino group takes place and the other amino acids are formed through transamination. The enzyme transaminase catalyzes all such reaction. For example, the two most important amide, asparagine and glutamine, found in the plants are as structural parts of proteins. These are formed from two amino acids, namely aspartic acid and glutamic acid, respectively, by addition of another amino group to each. The hydroxyl part of the acid is replaced by another NH2 radical. Since each amide contains more nitrogen than the amino acids, they are transported to the other parts of the plants via xylem vessels. In addition, along with the transpiration stream, the nodules of some plants, example soya bean, export the fixation nitrogen as ureate. These compounds also have a particularly high nitrogen to carbon ratio. Thank you.